Welcome to today's science lesson. If you remember, in the past lesson, we were dealing with the sexual reproduction in plants. We shall continue with the sexual reproduction in plants, and when we are through, we shall start also to see asexual reproduction in the plants. When dealing with sexual reproduction in plants, we had seen the part of a flowering plant. We had seen the parts of a flowering plant that are important when we are dealing with sexual reproduction in plants. We had seen the various parts and their role they do play. We had seen the male part of a flower plant, the female part of a flower plant. We had seen a symbol diagram of how a flower looks like. When we were still on the sexual reproduction, in plants, we came across the term pollination. And when we were talking about pollination, we covered two types of pollination. We mentioned the first type of pollination being the self-pollination, whereby pollination takes place within a flower. A flower itself has the male part and the female part, and pollination happens. What happens is the anthers is able to produce the pollen grains, and within the same flower, that pollen grains move from the anthers to the stigma of the flower. And we describe such a situation as self-pollination. But we went ahead and mentioned cross-pollination. We said cross-pollination is a situation whereby pollen grains from the anthers, they do not carry out pollination just within the same flower. No. Pollen grains within the anthers are transferred to another flower within the same plant. Are transferred to another flower within the same plant. We say that type of pollination is still self-pollination because it is within the same flower. But we said when the anthers of a particular flower, they go and pollinate the stigma of another flower plant, different from this plant, of the same family, of the same kind of family, and of the same family of the same kind. We say that one is cross-pollination because the pollen grains from another of one flower has gone and pollinated a different flower of another plant of the same kind. So when we are dealing with the pollination, we say there were two types of pollination. We had covered this self-pollination, and we covered the cross-pollination, where we said cross-pollination is the transfer of pollen grains from the anthers of one flower to the stigma of another flower on another plant, but the same kind, the same family. Now we want to deal with agents of pollination. For pollination to take place, there are agents that facilitate this pollination to be easier to happen. So agents of pollination. Agents of pollination. We know pollination will assist in the fertilization and reproduction of new plants and so forth. The first agent of pollination we can talk about is the animal. The animal. And when we talk of the agent of pollination being the animal, we say these animals that can carry out pollination within flowering plants are, one, the birds. The birds, they fly, they are attracted by the good smell, that is the scent, and you'll find they will move from one flowering to another. When birds are moving from one flowering plant to another, within their feet and on their feathers, they can carry pollen grains from the anthers of these particular flowers to the stigmas of different plants, of other plants of similar kind. So we are saying that it can be done by the birds, also it can be done by mammals. Mammals are agents of pollinations, and also insects. Also, we have insects. And when we mention insects as agents of pollination, here we can specifically mention the bees and the butterflies. So we say the bees and the butterflies can carry these pollen grains from the anthers to the stigma of within the same plant or within the different flowering plants. We know bees will always go to this flowering plant with the need of collecting nectar. So when they want to collect nectar, their wings and their legs will carry the pollen grains from the anthers of that particular pollen gland, and they will be transferred to the stigma, and later on fertilization will take place. When we talk of butterflies, they can be attracted to this particular flowering plant maybe because they have a bright color or they have a good smell. That's a scent. Scent can attract these butterflies, and as they move from one plant to another, they will carry the pollen grains, and the pollen grains will find its way one way or the other to the stigma. When they reach the stigma, they will find themselves to the ovary and later on to ovules, and fertilization will take place. So we have seen animals being one of the agents of pollination. The second agent of pollination is wind. Wind normally, for pollen grains to be transported by wind, they will need to have some qualities. These pollen grains that are manufactured by others, they need to be a bit light so that wind can carry this pollen grain from one flowering plant to another flowering plant. So when we go to the characteristics, such a pollen grains then will need to be light so that wind can easily carry them from one plant to another. So wind is another agent of pollination. The next agent is water. Water can carry pollen grains from one end of the river to another. And if they do this, then they will cause what you call pollination, and fertilization will occur. So these are the three main agents of pollination in flowering plant. We can try to see the characteristics of the insect pollinated flowers. We can compare them with wind pollinated flowers. We want to know the characteristics of wind pollinated flowers, flowers which the agent of pollination is wind. How are they adapted? How are they adapted themselves so that they can be transferred from one end to another by use of wind? Then we shall see how insect pollinated flowers have adapted themselves so that the insects can carry out the activity, that action of pollination. Therefore, we can have something similar to a table where we can have insect pollinated flowers, and then we can have wind pollinated so we have insect pollinated flowers this side and wind pollinated flowers. We want to see the characteristic. One insect pollinated flowers, they are brightly colored. So you find such flowers are bright. They are bright to the extent that the insect can be able to see them from far, and they can get attracted to the bright color. With this other one, they are dull colored. They are dull colored, therefore we cannot talk of so we find that insect-pollinated flowers are brightly colored, while the wind-pollinated flowers are dull in color. So there's no whether they are colored or not. Wind will not depend on the color to carry out its activity of pollination. No, still the pollen grains will be able to be carried from one plant to another. Then the second characteristic, the insect pollinated flowers are large in size. They are large in size, and this other end, wind-pollinated flowers, they are small in size. So we are seeing that characteristics when we are comparing the insect-pollinated flowers and wind-pollinated flowers, that characteristics with insect-pollinated flowers, they produce nectar. So they have the nectary at the base, the base of the ovary and so forth, where we have the sepals meeting. So they produce nectar, while the wind-pollinated flowers do not produce nectar. The fourth characteristics of insect-pollinated flowers is they have strong and nice smell. They have the scent that attracts these insects. So we're saying they have strong, nice smell. So when we say they have a strong, nice smell, they are scented. Then the wind-pollinated flowers do not have smell, not scented. Then we can advance to the fifth characteristics of insect-pollinated flower. They have small anthers firmly attached to the filament. They have small anthers firmly attached to the filament. Small anthers firmly attached to the filament, and then they have large anthers loosely attached to the filament. Loosely attached. So we can say that when it comes to the anthers, wind-pollinated, the anther is small and is firmly attached to the filament. But when you go to the wind-pollinated flower, then we are being told here they are large. The they have large anthers which are loosely attached to the filament so that they can easily be blown off by wind and carried nicely to the next flowering plant. Then we have another characteristics that differentiates the insect pollinated flowers and wind pollinated flowers. They produce fewer and heavy sticky pollen grains. Fewer and heavy sticky pollen grains. The few heavy and sticky, the sticky pollen grains so that once the insect passes by, they will be able to stick to that particular insect so that it can move it to the stigma and so forth. So they produce fewer heavy sticky pollen grains. The other side, they produce many light powdery pollen grains. Produce many light So we see that at the end, the wind pollinated flowers, they produce many light powdery pollen grains. Yes, they produce many light powdery pollen grains. Then we can have the seventh characteristics. The stigma is found inside the flower. The stigma is found inside the flower, while the other end the stigma hangs. The stigma hangs outside the flower. The stigma hangs outside the flower. We have seen those seventh characteristics. We can have the last 8 characteristics. The other characteristic here, the insect pollinated flowers, they have heavy and sticky pollen stigmas. Heavy and sticky, they have heavy and sticky pollen stigmas and pollen grains. They have heavy and sticky stigmas and pollen grains, while the other side we have heavy, they have large, hairy or feathery stigmas. The large, hairy or feathery stigmas. So we have seen close to 8 characteristics of insect pollinated flowers and also 8 characteristics of wind pollinated flowers. They have adapted in a one way so that wind can find it easier to carry out pollination, to carry the particular pollen grains from the others to other flowering plant. Now, we can have another when we want to deal, we want to know exactly what transpired, what transpires when now the pollen grains develops a tube and goes down to the ovary, then the pollen grains are able to combine with the ovules, what transpired. Therefore, you can have subheading fertilization in plants. All of these pollination activities that are taking place so as for fertilization to occur. We shall start by saying what is fertilization. We can describe fertilization as the fusion, in bracket, the fusion or the union between the nuclei of a male reproductive cell, that is the pollen, and the female reproductive cell, that is the ovule of a flower. Therefore, we can have a simple definition of fertilization. Fertilization is defined as the fusion bringing together the union. The fusion is the fusion between nuclei. Between the nuclei of the male reproductive cell which we can call it in this case the pollen, the male reproductive cell, and in this case is the pollen, and female reproductive cell, the ovule. And the female, it is the fusion between the nuclei of the male reproductive cell, the pollen, and the female reproductive cell, the ovule of a flower. That is the definition of what fertilization is the union of nuclei of a male reproductive, that is the pollen grain, and the female reproductive cell, that is the ovule of that particular flower. Now, we want to see what happens during this process of fertilization. Now, we want to have an explanation of what occurs exactly during fertilization process. One, the pollen grains falls on the stigma and germinates and forms a pollen tube. So the pollen grain falls on the stigma, germinates, and grows. It grows and forms a pollen tube. So that is what happens. Immediately the pollen grains is produced by the anther. Then when it falls on the stigma, it is able to germinate. And when it germinates, it grows a pollen tube that goes down through the style to the ovary. Then after that, we are on the process of fertilization. The second activity that takes place, we say the pollen tube grows from the stigma into the style and into the ovary. So we say the pollen tube grows from the stigma into the style. Yes, it grows from the stigma into the style and into the ovary. Into the lastly into the ovary. And what we know about the ovary contains the ovules. The ovary contains the ovules, and when these ovules combine, or when they meet with the pollen grains, then we say fertilization has taken place. So we are saying the next action, the next process that takes place after the pollen tube grows from the stigma to the style and then into the ovary, what happens on the third? Once inside the ovary, the male sex cells that are carried by the pollen tube fuse with the female sex cell. Inside the ovary. Once inside the ovary, the male sex cells, that is in bracket, the male sex cells, that is the pollen, fuse with the female sex cell. They fuse with the female sex cell in the ovule. They fuse with the ovules, and we call this process called fertilization. So once the pollen grains reach the ovary, then they enter that ovary, they reach the ovules. These pollen grains will fuse with the ovules. When they fuse with the ovules, then we call that process fertilization. We say now fertilization has occurred. Now, what happens? What happens after fertilization? What happens after fertilization? What happens after fertilization? One, the ovary grows to become a fruit. So what happens after fertilization? Once the fertilization has taken place, then you remember the ovary. The ovary contains the ovules. The ovary contains the ovules. So we are saying the ovaries will grow and become a fruit. That's okay. Then ovules within them will become seeds. Two, the ovules are within the ovaries will grow to become seeds. So we're seeing the ovary will grow into a fruit, and maybe when you check within the fruit, then you're going to get the ovules, which now will grow into seeds. Then What are the actions that happens after fertilization? We say the petals. If you remember the petals, the petals, the style, the petals, the style, filament and others. The petals, style, filament, others, and stigma. And the stigma all shrivel, and the stigma, we are saying all shrivel, dry, and fall off. So we are saying that once the fertilization are taking place, now what roles now is there for the petals, whether they are corals, calyx, what functions are there now? We are told the style, filament, others, and the stigma all shrivel, they dry, and they fall off. We are told a scar is left by the dry style. A scar can only be traced. A scar is left by the dried up style, a dry style. Then the fourth actions that happens after fertilization, the flower stalks becomes the fruit stalk. We had talked about the flower stalk, that is that bud, that base bud that is able to hold the flower. Now that flower stalk now becomes the fruit stalk because now the ovary has changed into a fruit. Now the flower stalk now will become the fruit stalk because now it will be holding the fruit. And we know within the fruit we have the seeds. This is the point of attachment of the fruit to the plant. This flower stalk, that is the point of attachment to the fruit of the plant. That is the flower stalk. We can easily now look into what transpired parts of a seed. What is a seed? Let us look into a seed. We have seen how seeds are formed. We have said after fertilizations, the ovules that are able to fuse with the pollen grains, they later on grow into seeds. We have said the ovary will grow into a fruit. Now we are dealing with the seeds, and we know these seeds were the ovules in the ovary. After fertilization, they became seeds. So parts of the seed. Now, when we are talking about a seed, we are interested in a seed that are found within the flowering plant. So in a flowering plant, a seed is usually found inside a fruit. Usually found within the fruit. That is mostly the seed is found within the fruit. We can have a simple diagram to illustrate what a seed is. And when we are talking about seeds, we have two types of seed. We have a dicot seed, meaning dicot seed has two parts. If we talk of a tester, has got two cotyledon, we shall come there. So it has got two seeds. We have the dicot seed and the monogot seed. We can just draw to illustrate the differences so that they can be visible between a dicot seed and a monocot seed. We can start with a dicot seed. We can have such structure whereby we say outside here, that is the tester, being the outer covering of a seed. And we can use this example. We can assume this is a bean seed, because a bean seed is a dicot seed. So you can have an example of a bean seed. Then maybe when you go to monocot, we can have an example of a maize seed. A maize seed is a monocot. So if we take this one as a this organ below there is a hilum, the hilum. When you look at the scar, we shall see the function of this hilum, what it does to that particular seed, and then a micropyle, and that is the micropyle. Then the cotyledon, cotyledon. Now, those are the parts of a dicot seed, but we can go further because we want to show what part grows into a shoot, the plumule, and what part grows into a root, that is the radicle. We can have further the following so that we show the dicot. We are what we want to emphasize. We can have this one as the tester, and then we can have these two parts as the cotyledon. We can have this one as the radicle, it's growing into a root. The radicle. We can have this one as the plumule. What happens to the plumule? You can see it will grow into a shoot, while the radicle will grow into a root. So if we observe keenly, these are what we are showing. These are parts of a bean seed. These are parts of a bean seed. We call them the dicot seed. It has two. So above we can see we have tried to show the parts. We know once the bean seed starts germinating, and we know what happens. The bean seed is made in such a way that it has an entering where it can allow some oxygen to come into, and it will allow oxygen to come into and water so that germination can occur. So we have seen parts of a seed, and we are seeing the parts of a seed we have seen. This is an example of a bean seed, and it is a dicot seed. It has two cotyledons, as we have indicated. Now let us go ahead and see what happens when we are seeing parts of a maize seed, and we know a maize seed is a monocot. It's a single seed, and we shall also show the parts that are relevant to the maize seed, the monocot. We can say yes, this is an example of a maize seed. If this is a maize seed, then we can have this as the radicle, and the other one as the plumule. So we know that the radicle will grow into a root, where the plumule will grow into a shoot. We can have this larger part as yes, we have the tester. Tester is the outer covering. Then we have the endosperm. Endosperm. The endosperms acts as a storage of food. This is the storage of food that will be able to support the germination of the plumule and the coming up of the shoot, that is the radicle. Then we have our cotyledon. The sperm, plumule, cotyledon, and radicle. So the above these parts so we can see it has got one coat, it's a monocot. This is the structure of maize seed. That's how it looks like. Maybe we have been writing these parts. We can discuss further the parts and functions of a dicot seed. Parts and functions of a dicot seed. So if we remember the parts, we can start with the first part, which we are calling the tester, those who can remember. And we will want to know what is the function of the tester. So where is it located? It is the outer part. It is the outer part, and it's also called the seed coat. It is the outer part of a seed, and it is also called the seed coat, which protects the inner part. So the tester, that's the part of the seed. It is the outer part, is also called the seed coat, and it protects the inner parts of a dicot seed. Then we can go ahead to see there's a part we call the hilum. We want to know the function of hilum and what role does it play in a seed. So the hilum, it is the point of attachment to the fruit. So you're saying hilum is the point of attachment to the seed, to the fruit. The seed is found within the fruit, that is the hilum. Then we can go ahead to the cotyledon. And we want to know the cotyledon and what are the functions of this cotyledon. They store food in dicots, not in monocots. So they serve as storage of food. They store food in dicots, and they're also called seed leaves. So they store food in dicots. So we are seeing the major function of the cotyledon is to store food that will support the growth of the plumule and the radicum. Then from the cotyledon we have for this the plumule. What are the functions of plumule? We are told the plumule, the plumule grows into a shoot. And we know what the shoot will become. The shoot will become that particular plant, the big plant that we have, the big plant, the big flowering plant, were once a seed and they germinated. When they were germinating, they had a plumule that grew into that particular plant. We can see flowering plant. So that's the work of the functions of plumule. Then the fifth, we have the radicle. And on the radicle, we are told this is what grows. They grow into a root. They will become the rooting system to support that particular flowering plant. Then from the radicle, we have the micropyle. What is the main function of the micropyle? One thing with the micropyle, it allows air, and specifically air called oxygen and water, to enter the seed. So it allows air, and we have said this particular air, oxygen and water, to enter the seed. So it is very important for that particular seed to be aerated. Oxygen is not only important when we talk about animals, it's also important when we're talking about plants. And the seeds have to be aerated to get enough supply of air that has oxygen. So it allows air, that's oxygen and water, to enter through the seed. Then we have the seventh part of, call it the embryo. And with the embryo, it consists of the plumule, the radicle, which together grow to become a new plant. It consists of the plumule and the radicle that grow into a new plant. A new plant is made from the plumule, a new plant is made from the radicle. Both of these is what we are terming as the embryo, and they grow into a big flowering plant that has stems, leaves, and so forth, flowers, and has roots. Roots are there to ensure that they support the plant and so on. Note, important point to note when we are talking about these functions. An important point to note is the part called the endosperm is known for storing food. Food in monocot seeds. So endosperm is known for storing food among the monocots, only among the monocots. Now from here we can try to see the differences that are there, the differences that are there between fruits and seeds. What differences can we tell? What differences are there that can be observed between fruits and seeds? The differences that are there between fruits and seeds, one, we can have a slide table, we can have fruits, and we can have seeds, so that we look them further. One, when we talk of fruits, they have two scars. The fruit scar and the style scar. So we are saying, when you want to see the difference between the fruit and the seed, we are saying, with the fruits they have two scars. One of the scar is the fruit scar, and the other one is the style scar. We know the style scar comes up after fertilization and it dries up when the ovary is developing. When you go to the other end, has only one scar, the seed scar. One scar, the seed scar. So we can say that is the first difference when you are comparing the fruit and the seed. Then another difference that is observable, fruits are often large, and they contain seeds. Fruits are often large and they contain seeds. So within a fruit, you can be able to locate seeds within them. When we look about the seeds, we are saying the seeds are often smaller and are contained inside fruits. We say are often smaller and are contained inside fruit. So we are seeing that the fruits are large, and within them you can be able to get seeds, and we are saying seeds are small compared to fruits and are contained within the fruit. The third difference that we can mention, they are the result from fertilization of ovary. Of ovary, meaning they grow from ovary. A fruit grows from an ovary, results of a fertilized ovary. The other end results a fertilized ovule. So we can say the fruit emanates from an ovary, while a seed comes from a fertilized ovule. Then the fourth difference worth mentioning, the outer covering of a fruit is called a pericarp. Yes, the outer covering is called a pericarp. It's called a pericarp, while the outer covering is called tester. Tester, that's A. Yes, the outer covering is called a tester or a seed coat. So we can see there's also a difference that comes to the outer covering. The outer covering of a fruit is called a pericarp, while the outer covering of a seed is a seed coat, or we call it a tester. So we have seen, up to now, we have seen how we have seen the differences between the seeds and fruit. We have seen the parts of dicot seed. We have also seen the parts of a monocot seed. We have seen the functions of these relevant parts of seed and so forth. We have seen the process of fertilization, what happens when fertilization takes place. We have seen, up to now, we have seen up to the seed. Now, what we can ask ourselves, from these seeds, how do they grow into plants and so forth? The seeds are able to grow into plants when they germinate. So we have a process that we call germination. So germination is the growing of seeds into new plants. Germination is the growing of seeds into new plants. These flowering plants, they do germinate. They germinate from what? They germinate from seeds. When seeds are planted, they are able to germinate. The plumule will shoot up and the radicle will grow into roots. The plumule will grow into that particular plant. There are conditions that are necessary for germination to occur. Just to mention but few, the important condition for germination to occur, or for that particular seed to change and to turn into a tree, to become a seedling, one condition is there must be oxygen. Now, when we talk of seeds, how do seeds grow? What do seeds become? When we talk of planting, you don't plant a fruit, you plant a seed. So when seeds are planted, they will undergo a particular process. They will germinate. What is germination? We are saying germination is the growing of seeds into a new plant. The growing of seeds into a new plant is what we say is germination. And we are saying there are conditions that are necessary for germination to take place. And these conditions are worthy mentioning. Condition number one is for a seed to be able to germinate, there must be air. and that particular air must have oxygen. In that air, what is important element is the oxygen component of it. So the seed, for the seed to germinate, it requires oxygen. Condition number 2 is water. And water should be available in what form? Water should be available in moisture. If water is available in the form of moisture, then that seed is ready to grow. The third condition that is very important for that seed to grow is the temperature, warmth. If the condition is very cold, then that particular seed might fail to germinate. So warmth, the temperature should be conducive and appropriate for that particular plant to grow. We are going to stop here, but in our next lesson, we are going to have close to 10 questions. The 10 questions will try to test us, our memory. Have we understood what reproduction in plants is all about? Therefore, we shall have some questions to test our ability as far as reproduction is concerned. Then after that, in our next lesson, we shall start on asexual reproduction in plants. Remember, today we have covered sexual reproduction in plants. Now, in our next lesson, we shall cover asexual reproduction in plants. Reproduction without the use of seeds. Thank you, and that is the end of our lesson.